All right. Please welcome our next speaker, Dr. Robert Oostenveld.
Okay, thank you for having me here, and also welcome to all the people that are following this online. What I want to do is I want to present in a rather different fashion than the presentations that we've seen yesterday. In preparing for this workshop, I've been more reflective of how I think that multimodal research is progressing. And what I will not do is I will not show nice figures with brain data and with significant effects. I will actually also not show any sophisticated data analysis methods or any explanations how to do single or multimodal data analysis. Rather, what I will do is I will start with a short description of my background and my expertise, which I think clarifies the view that I subsequently will share on multimodal analysis. And I will give an assessment of my successes, but also of my failures. Because I think that from looking at our failures, we can learn in which direction we should be heading in the future. And I will try to make a critical evaluation of where it goes wrong and where it succeeds. And I hope that it is going to become more clear during the course of the talk. So this is me sitting there, my head being scanned. It's actually the EEG electrodes on my head being scanned. This is during one of the field trip workshops. So field trip is one of these projects that has shaped me considerably over the last 15 years. It's something that I started shortly after my PhD. Field trip is this Butler-based toolbox for MEG, for EEG, for intracranial EEG, local field potential spike, and also recently NIRS analysis. And the development of field trip started in 2004, which is more or less shortly after I obtained my PhD and at the time that the Donors Center started in Nijmegen. In 2011, we published a reference publication, which is now cited almost 4,000 times. Let me just see whether I can move this one out of the way. Yeah. And as far as I can count, although the things that Alex showed in terms of counting, they were much more informative. So I actually also looked them up for field trips. It's fun to see the detailed stats that you can get online. But as far as I can tell, there's about 50,000 times field trip has been downloaded, excluding GitHub and excluding redistribution by others. And I think that there's about between 3,000 and 5,000 people that are starting field trip, let's say, on a weekly basis. I've been able to identify like 90 people that have contributed code to field trip. And I think it's very relevant to mention that field trip is also being used by packages which have been around longer, EG Lab and SPM. And so we're working actually quite closely together with different open source tools, also with the MNE Python, as Alex already showed. So we're actually sharing a lot of the methods. And as such, we're not really competitors, but we strive to achieve the same goals. However, field trip is way more than software. Field trip is also a way of disseminating and bridging knowledge. So over the years, we've organized more than 60 training workshops. We're organizing one every year in Nijmegen, but we're also organizing them at many other locations. And we have a user community which is centered on the mailing list, which has about 1,500 active, 1,500 people on the list. And we have a wiki-style website, which has more than 1,000 pages of documentation. So tutorials, examples, frequently asked questions, and all that type of stuff. And with field trip, with the project as a whole, so not only with the code base, we're also trying to provide a bridge towards the rest of the MEG or the EEG community that are developing methods, which also includes companies. And I'm really glad that we have contributions from various companies to the code base, and companies also relying on field trip being valuable for their stakeholders, for the people that buy their equipment, because I think we really rely on companies also providing the tools that we're using in our labs, because we cannot develop everything ourselves. So having an inclusive ecosystem is very important. Something else that over the years has shaped my view on neuroscience is the Donner Center. So this is a photo of the Donner Center. My room is actually just outside of the photo. And at the donor center, we do cognitive neuroimaging. So we have 3 MR scanners on site. We have an MEG system. We have EEG labs. A lot of people are confused about this donor. So the donor Center for Cognitive Neuroimaging is part of the Donors Institute for Brain Cognition and Behavior, which is part of Radboud University Nijmegen. So as such, we're all on the same campus. We often call it donors because it's just an easier name than Radboud. At the Donor Center for Cognitive Neuroimaging, we're not so much studying biomarkers, biological mechanistic models, or data-driven models, which have been presented in great detail yesterday. Although we have some researchers such as Christian Beckman, who's definitely into data-driven models. But rather, I would say we're using neuroimaging methods to understand cognition in the healthy brain. And this is important to keep in mind because in the rest of the presentation, I will occasionally make general statements about how I think that the field is working, but it primarily relates to my field. So that relates to cognitive neuroimaging in healthy subjects. So if I look at the modalities, the data modalities that we acquire during experiments, and especially during cognitive experiments, then I would split them in a few categories. On one hand, we have physiological signals, MEG, EEG, fMRI, nearest EMG, but also heart rate and other signals. We have a lot of behavioral measurements, such as button presses, eye tracking, motion tracking. Sometimes we have video, we record speech, and very often almost always we record these simultaneously. And although an EEG experiment with button presses, typically people wouldn't consider that a multimodal experiment. I think we should also see behavior as a relevant modality that we can record and that we can study. And then we also have time-invariant data, such as MRI, like anatomical MRI, T1-weighted images, diffusion-weighted images, but also things such as questionnaires, but of course also genetics. And if I start from the modality that I'm most comfortable with, which is EEG or with MEG, then this is typically how, for me, such an analysis would look like. So I would take the data, I would start with time series analysis, do spectral analysis. I will make a volume conduction model on the basis of anatomical MR scan. I would construct a source model. I would estimate biophysical model parameters, such as dipole positions or distribution of source strength throughout the brain. I would come up with a statistical model, for example, as depending on the experimental research question, of course, for example, multifactorial experimental design with multiple levels. And I would link this to a cognitive model in which I would map this onto different cognitive processes that we're studying. However, if I start thinking about button press data, so this puzzled face, that's me. Because actually, we have button press data. I know I can compute reaction times, the percentage correct, but that's pretty much it. So I realize that I'm very limited in interpreting this data, whereas I know that there's research fields where people make very sophisticated analysis of behavioral data recorded from a single button press, from a series of button presses. Of course, I can still take my very simple analyzed reaction times. I can put that into a statistical model, and I can try to relate this to a cognitive model. The same also holds for eye tracker data. I typically will be using eye tracker to just check whether the subject is looking at the fixation cross where I told them that he should be looking. I'm not doing sophisticated analysis on the eye tracker data. I might be doing some saccade analysis, but I realize that I'm very limited in my understanding of these modalities. But then also, if I look at other people, so the people that I encounter at the donors, the people that I encounter at training events, I realize that they have the same positive look regarding the EEG or MEG data. And that brings me to the following idea. So we have data from different modalities. We have, for example, MEG data, fMRI data, button press data, eye tracker data. And for each of these, we have, within specialized domains, we have processing pipelines. We're doing an analysis, and if I'm thinking about multimodal analysis, Then I think the idea is that we go from the physiology, which is related to the signals that we're recording, to a joint data model analysis, which is more at the level of the brain. Then we go to a cognitive model, which is related to the level of the cognition or the mind. And so these are the different domains, but these are also the different silos that we've already been discussing yesterday during the panel discussions, that there's actually quite some distance between these different silos. And what I realize is that very often people, including myself, actually revert to skipping the joint analysis model and just try to go as quickly as possible to a p-value. So rather than there being a sophisticated data analysis model, or rather than there being a sophisticated cognitive model, the pipelines are developed in such a way that we can get to a p-value. And I realized that this is something that I've been contributing to myself as well. So in 2007, Eric Maris, a colleague of mine in Nijmegen, and I wrote a paper on cluster permutation statistics applied to EEG and MEG. And this is a very successful paper. It cited a lot, the method is used a lot. And the basic idea is that we start with the notion that a randomization or a permutation test allows the use of any statistic, any test statistic. So the idea is that we have implemented is that we can convert a massive univariate statistic into cluster mass. And then using a Monte Carlo approach, we can use a randomization, we can compute a randomization distribution of the cluster mass, and we can use that to assess the probability of the largest cluster mass as it was observed in the original data. So that gives us a chance of testing a hypothesis of computing a p-value. And the two hypotheses that we're considering is the null hypothesis that the data is exchangeable between conditions, and the alternative one that the data is not exchangeable. Hence, the data in the conditions is different. And what I want to draw your attention to is the very limited information that we're getting from the alternative hypothesis. By just saying that the data is different, actually we don't know which feature in the data is different. It's a non-parametric test, actually doesn't even relate to a parameter of a model. So this is actually a test approach which is extremely sensitive, but also extremely non-informative, because it doesn't inform us about a model. So if I think on how we've been developing over the years with field trip, so starting around 2003, 2004 with MEG data, in 2004 we started using LSP data, intracranial invasive recordings in animals. We added EEG, we added intracranial EEG, we added NIRS, and we're still working on new modalities as well. For each of them, we're basically making the same mistake, is that we're trying to jump to a p-value as soon as possible. And that also makes it attractive to integrate all of these methods within the field trip toolbox, because we already have the statistical machinery. So it's actually very simple to use the same cluster-based method for NIRS data or for intracranial EEG data, etc. So that's, as a development model and towards happy customers, that's very nice. But the thing that we're skipping is this joint and data model analysis and these cognitive models. So what I see in reflecting on like how I've basically been contributing to the field of multimodal imaging is that we have a lack of joint statistical models for data. So if we do statistical modeling of multimodal data, we can analyze data from, let's say, 40 EEG subjects or from 40 fMRI subjects, and we also can analyze 40 combined EEG fMRI experiments just fine. But what if we record both modalities from different subjects? Then it already starts getting difficult, because we simply don't have very rich statistical models that allows us to integrate data from different subjects. Or what if we use slightly different tasks? So for example, if we study language processing, but we're recording language processing either from auditory for the visual domain, and all of a sudden the statistical model become much more complex. And what it ends up in is that many studies have multiple p-values that shape the discussion of the publication, one for the EEG and one for the fMRI. As a strategy, it makes a lot of sense, like to divide and conquer, but I think it lacks the opportunities that we really have with multimodal imaging, because that requires a joint model. Thinking further about this and also discussing this with colleagues in NIMH, I think we also have a lack of joint physiological models for data. Oh, by the way, I should add to this that of course yesterday Vince showed a very nice way of how to do data-driven approaches which do integrate different types of data. So I'm not saying that this is in general not possible. I'm just observing that in the environment that I'm in, so this environment shaped by field trips, shaped by the donders, I see a lot of people that are not taking it to the level of a joint model. Okay, so I also see this lack of a joint physiological model for the data. So we have relatively limited understanding of the physiological signals. We don't, for some of the types of data that we're acquiring, the understanding of the relation between the signals is not clear. And the interaction between the signals and behavior and cognition is also not really clear. And therefore, a lot of the researchers are using correlational models or correlation analysis. We have very few mechanistical models, although I should say yesterday we've also seen with the virtual brain project, and I think DCM is also a really nice example of dynamic causal modeling using generative models, that there are attempts at making physiological models to explain data and to use this in science. But actually in my environment, these are not getting a lot of attention. Most people are doing correlation analysis. Okay, so far I've been relatively critical towards myself and towards my environment. But I think it's also fair to give a few positive notes. So if you look at the different types of data that we're processing a lot within Fieldtrip, I would say that we have been able to successfully bridge the gaps between different containers, between different research domains by bringing these together within one unified project where people can learn from the different modalities. So people benefit from the knowledge that has been acquired in different modalities. Another thing that I think is really important is that over the last few years we've spent a lot of time, like as a community, work on BITS. BITS is the brain imaging data structure, and it was already shortly mentioned yesterday, and Alex also mentioned in this morning. And what I want to do is I just want to give a very short intermezzo about BITS for those of you who don't know it yet, because it's relatively new, and I think it's really important that we continue moving in this direction. So what is BITS? It is a simple and an intuitive way to organize and describe data from MRI, from MEG, EEG, intracranial, and behavioral experiments. BITS allows you to use tools, so-called apps, to facilitate the analysis of larger data sets. And the standards, so the description of the specification and its updates are published on this website. A good introduction to the BITS standard can be found in an initial paper which was published in 2015, I think, which describes BITS for MRI. And since then, we have extended the standard with separate papers also for MEG, for EEG, and for intracranial EEG data, which also have appeared in scientific data. So there's now multiple papers. Each of the papers has to be relatively short, of course, like with this page limits. And I would say that the online documentation is more complete and more detailed. So if you want to get an overview, you can look at the papers. If you want to know how it works, you should look at the website. So why would BITS be relevant for you? BITS allows you to organize and document your data for sharing. And sharing is not necessarily sharing with the rest of the world. Sharing is first and foremost with your future self. It's about documenting data such that within 1 week, 1 month, 1 year from now, you still understand what you have been doing in the lab. It's also documenting your data with your current and with your future colleagues. Because often we acquire data that will be used by others, and often people start with data that was acquired by others. And of course, BITS is also about sharing with the community. And then that's where we have to consider legal and ethical constraints on the type of data that we're acquiring. If you organize your data according to the BITS specification, then you're organizing such that you can use more efficient analysis tools. So FieldTrip is one of these tools, but also EEG Lab, Brainstorm, or M&E Python are tools. SPM, there's also tools like pipelines built on top of FSL. OpenNeuro is one of these platforms where the data and a lot of these tools come together. I already mentioned that we have BITS apps, but we also have BITS MATLAB, which is a library to more easily work with BITS data sets in MATLAB, and we have PyBits, which is doing the same thing in Python. And these help method developers, or these help researchers to very quickly query and use large data sets. Okay, so a question I often get asked is like, so BITS, that's a new standard, and I think a lot of you know this coming from XKCD. So we have a lot of standards, and then of course we complain, like none of the standards is really suiting our needs, so we define a new standard. And then the next situation we have N+1 standards. I think it's very important to stress that BITS standard, BITS is not a standard. BITS is not a new file format. BITS is not a search engine or a data sharing tool. BITS is a collection of best practices that builds onto existing standards. So we're using existing file formats. I would rather say that BITS is a hands-on practical solution for organizing large data sets, but it's more a convention. It's something that we jointly can agree upon. If everyone follows this convention, then our research will be much easier to conduct. And to just give you an example of a multimodal data set, so this is a directory organization of a BITS data set. And what you can see is that there's a directory structure and there's a certain file naming hierarchy. So over here you see the directory structure, which organizes data per subject and then per data type. In this case, anatomical MRI, MEG, EEG. Over here you can see the actual EEG data. So BITS specifies a limited number of file formats. So for MRI it's NIFTY, and for EEG it's a brain vision format, or it's EDF, or it's EEG Lab format. So those are the formats that are allowed within the BITS specification. But perhaps more importantly is that there's metadata. There's both metadata at the data set level, there's metadata at the recording level of the individual subject, where we store information about amplifier settings, but also store information about the participants, for example. This metadata is stored in TSV and in JSON sidecar files, and for example includes information about the participants, it includes information about the equipment, it includes information about the cognitive task that the participants were engaged in, which is typically one of these things which is not recorded by our equipment. Like we record trigger codes, but we don't record the instructions that were given to the subject. And there's also some details from the files itself to make querying easier. And just as an example, so this is for an EEG data set, this is the channels.tsv file. It just has a few required columns, such as what's the name of the channel, the type of the channel, the units. You can add extra columns such as the status or a status description, or the description of electrodes, which are not channels like electrodes, so they're small discs that you attach to the scalp, whereas the channels are part of the amplifier. So also electrodes have a name, and you can record a location for the electrodes. And if you record a location for the electrodes, you have to specify in a JSON file, it's the quad system JSON file, how those coordinates are to be interpreted. But it means that rather than having to guess what these numbers mean, you can actually look up that these numbers are to be interpreted in millimeters, that the origin is halfway between the pre-auricular points, and that the positive x-axis towards the right. And this helps a lot in the very diverse data that we're recording. So rather than saying that everyone should converge onto one coordinate system, what we're doing is we're just describing the coordinate systems that are in use. So the outlook with the BITS project, which is a very active project with a lot of contributors from very different domains, is that we want to extend its adoption. We want to extend the supported data types. So genetics is actively being developed. PET-CT, but also anatomical MRI with multiple contrast is actively being worked on. There's efforts to get eye tracking in BITS. There's some discussion on getting NIRS in BITS. Multi-electrode arrays are being discussed. But I also think that an important aspect is what we're working on with the wider BITS community is derived data, which means the output of these analysis pipelines that I've been referring to earlier. And then for MRI, you can, for example, think about spatially processed data, or spatially transformed. Or for fMRI, it's model estimates. Or for electrophysiology, it could be pre-processing or source reconstruction. So these are things that we're currently discussing. So at this moment, this is only for raw data, but we want to extend it such that we can also have results analysis. So let's move back from bits again, back to my talk. So we have bits as a general structure that helps us to bridge the boundaries between these different silos, that we have a joint representation of the data. So we have open source projects such as Filter, but also such as MNE Python, which can serve to bridge between domains. And we have open data such as bits, which helps to raise awareness and facilitates research across modalities. But bits derivatives, so again, switching to a more critical note, bits derivatives for the archiving and sharing of process data and results is already being discussed for quite some time. And I have to say that we're not really reaching consensus yet. And I think that is partially due to us not really having consensus on how we want to describe this joint data analysis model. It is, I think for a lot of us, it's actually not so clear what the results are that we're working towards, except for these p-values that we're very often reporting. I think this is one of the reasons that the discussion on bits derivatives is going so slowly, because reaching consensus on something that we're not often discussing, well, that's difficult. So I would say that in each of the modalities, or in most of the modalities, and that's what you see once you start doing multimodal imaging, we have rather weak quantitative reporting. Like our scientific reporting is focused on statistical inference and reporting p-values, making decisions. But how should we report quantitative results? And how should we allow researchers from other expertise domains to integrate results? Or how should we allow other research to use the effect sizes that we have quantified? So this is still relatively immature, I would say, compared to how we're otherwise doing science. I also think we should think about how to make explicit predictions, like how to use the exploratory part of the research cycle in contributing to the hypothesis-driven part of the research cycle. So that's where I'm critical towards myself. If people use cluster stats on their EEG or MEG or whatever data, they're basically what they're saying, I expect the data to be non-exchangeable due to some evidence somewhere represented in a cluster in my data space. So that's very uninformative, which also means that if people make such a statement in their paper, it's very difficult for other researchers to build upon that knowledge. So I think one of the challenges that we have is that we have to do more quantitative reporting. And sometimes it's not that difficult. So we can report ERP effects in microvolts, MEG effects in femtoteslas, or after source estimation, we can report effects in nanoampere meter. But it's different than what we're now doing, is that we're showing source level results in p-values or in p-maps. And the same goes for oscillations, which we should report in proper units, and the same goes for latencies and time minutes, which we should also describe in proper units. I think we should, again, get more used to the quantitative aspects of our data, also on the reporting side. And if I zoom out a little bit, and this is something that that was already addressed to some extent in a panel discussion yesterday, is that I think that we have a suboptimal incentive structure. Research is to a large extent being done by junior researchers as part of their training. And it's in hiring young researchers, but also in assessing what they have achieved. It's quite difficult to assess interdisciplinary skills. It's much easier to assess whether a young researcher is mastering a specific field. So I also personally, I would feel more comfortable hiring someone who has expertise in MEG than someone who has expertise in communication or expertise in working together, because I don't know how to assess that. Individual researchers are motivated by career opportunities. Makes sense. That also goes for me. which means that first or last authorship papers, that's really the currency that drives individual researchers. It also means that we tend to optimize our research strategies for quick publications, which are relatively simple. So typically just like one paper per year for a PhD student or for a postdoc, because that's basically how you build up the currency. And I would say that we don't really have the incentives for team science. So there's this career opportunity of a jack of all trades or of a methods person. Doesn't seem to be too attractive at the moment, but I think that these people are really important for getting the multimodal research to a higher level. And the funding structures that we're relying on, they're mainly directed towards research producing the primary outputs, so to the PhD students and the postdocs. And I think it's important that we also implement incentives that contribute to the secondary research outputs, such as code, such as dissemination, such as training. And I will really start valuing that, because it is being built into grant schemes, but it's not really being used that much yet. And further at this level of zooming out, I think we also have to consider these boundary conditions under which we're doing the work. So we can and we should try to provide more training across individuals. But if the individual training aims to develop experts in a narrow field, which I think is also a really important characteristic of our academic training, then team science should also be employed. So and should be embraced. So we should also train people on communicative skills. Like as was already mentioned yesterday, like making sure that research actually understands research that are working in another domain, having a shared language. I think that's one of the things that with BITS, I really realized we had a lot of discussions with people from the different data modalities to actually understand what we mean with what's a scan. For an MRI person, a scan is very different, or might mean something totally different than for an MEG or for a NIRS person. I think it's important that we link existing tools over discipline boundaries, but also that we link the existing expertise that we already have within these domains. And as I already mentioned, I think it's important that we link data over boundaries. So not that we just define the standard for MRI data and then something else for MEG data and something else for EEG or for NIRS data, but rather I think we really should continue to put effort into making a single open data structure that allows us to capture, to represent multiple data from multiple modalities. So to summarize, I think that open science results in better collaboration and more awareness, and specifically open source, such as Fieldtrip, but also MNE-Python, as Alex has very clearly shown this morning. And on the other hand, that open data is also really important for this because it also brings together people from different disciplines, from different silos that have to sit together and to figure out how to bridge the gaps between their domains. Tool development, such as Fieldtrip, can span multiple domains and multiple modalities. But I think it's important that tools don't have to be monolithic. We don't want to have one software package that does research for us. I think bridging between tools is as important as bridging between research domains. So for me, one of the reasons to attend the CodePrint in Boston is also to hang out with the MNE-Python developers to understand what drives them, what their development model is. I have to admit, I didn't write a single line of Python code during that week, but I learned a lot. I think, so if we continue strengthening the links between the different tools, I think that's going to be very beneficial for multimodal neuroscience. And the voids between modalities have to be resolved by having people collaborate, by linking expertise over different labs or between different departments, different universities, and by providing training. I think we should be using team science for strengthening and for improving the individual incentives and to contribute, and we should try to get people to contribute to fields that are just outside their own expertise regions. Because I think that that's what's necessary to bridge these boundaries. Like each of us has our own expertise, and we all have to step outside of our own comfort zone into someone else's research expertise, and then we have to basically start interacting with each other. And I think if we're able to achieve that, then we can break the boundaries between these silos, and we can work towards a more efficient future for multimodal neuroimaging. Okay, thank you.
We have time for 2 to 3 questions.
Mine's a comment more than a question. When you were talking about some of the other more static modalities of data, and you mentioned genetics, it occurred to me after what Molly was saying yesterday with the work that she's doing across the lifespan, that we're going to have to also rethink that variable, because genes get expressed at during different developmental milestones, and they keep changing through the lifespan. And so that was something that I hadn't really connected until I heard your 2 talks.
Yes, I think that's a very, it's also, that's one of the types of data that I'm least familiar with. So I tend to put it in the bucket together with other static data that I'm not really in detail work on. Because also anatomical MRI, if we look at developmental studies, like developmental neuroanatomy, all of a sudden an anatomical MRI is not static. It changes, but it changes more slowly. It's a very different time scale than the time scale that I happen to be thinking of mostly.
This may be a little bit out of the box, but as we begin to accommodate multiple data domains in our models and our thinking, as you described quite beautifully in your talk, from a practical point of view, this raises a number of questions, one of which we haven't talked about in this workshop, and that is power analysis. And that is, as we start integrating issues like genetics, for example, into our data domains, and we want to ask questions like how these factors actually relate to the dynamics that we're studying, whatever it is, then we have to make estimates of the effect size.
Yes.
And I think this is a very important question, because how much data is enough to really answer or address your hypothesis? And I personally don't know how to deal with that, but I do know that I'm barraged with that question all the time. Yeah. I'd like to think about that a little bit.
Yeah. So this is something that I really recognize. Like in some of the larger projects that I've been involved in, such as the Human Connexome project, we basically just went for a lot of data, and that's also something that the people working on some data-driven analysis methods, such as Alex or Vince, they basically they would always say, please give me as much data as you can. But at the Dons, we're also doing developmental neuroscience, so we're working with babies, and then so all of a sudden you have a much more challenging population that you have to sample from, or we're working with patients, which is much more challenging to get enough subjects, and also it's much more of a burden for those participants to our studies. So I think we have to be critical also, is that we should not only try to acquire as much data as possible. We should be making fair assessments of how much data we need. But it's a question that we're very often discussing at the donors, and it's even a requirement for our internal review process that you have to justify the number of subjects that you include. But at the same time, we don't really have the right tools for it yet. And I think that's partially because we don't have the data that is required for the tools that are currently available because of lack of reporting.
Just make a comment. One of the ways to deal with that, I think, is to differentiate really hardcore hypothesis-based investigation from investigation that's exploratory. And I think this raises a really important point about how important some of these multimodal ventures are. And if we just present them as preliminary investigations, leaving perhaps issues of power for something, say, second stage, then it's a way to
something that I like in software development, and that's where Alex is really an expert in, for example, in versioning. In software, we're totally used to versioning software versions, and software versions tend to get better or tend to get more features over time. We're not really versioning our research outcomes. We're not really it's still relatively uncommon to version our data sets, for example. But I think that this continuous integration or expansion of our insights, we can link that to versions. And I think that also for our research mind, we should just have our first publication would be version 1.0. And if we then find a bug, we should just be willing to publish that as version 1.1. It's basically, it's a bug that has been fixed in a paper. And if the insight really changes, then we should just publish a paper version 2.0.
So I was triggered and very happy to see your comment about reporting things in the right units. I'm a little hoarse because I've been saying for a dozen years, complaining how important it is. And let me just sort of briefly stand on my soapbox for this. Reporting T or P values is about the least repeatable thing you could ever report, right? It depends on N, it depends on all kinds of jazz. And reporting the P statistics across a paper is a completely useless endeavor for replication or metadata analysis. Reporting 0 and first order statistics is absolutely what we should be doing. Percent signal change, I guess I will comment, femtotesla I don't think is a useful number because it actually depends on the proximity of the detector to the brain. And it's unnormalized. And there's no natural unit for MRI, microvolts per cubic something. But reporting in simple normalized units makes a great deal of sense. Reporting standard deviations actually still allows metadata comparisons across papers. And it's unbelievably and particularly important when we look at these multimodal data analyses because the idea of combining data across studies when you're only looking at oddly derived higher order things like p-values from probabilistic ICA, you simply can't use those to do any meta-analysis. Yeah. So I applaud that, underline it, scream about it, but as I say, I'm hoarse.
Okay, thank you.
Let's give another round of applause.